Hello and welcome to BioBiz Buzz, podcast on the science and business of biotech, pharma and medtech. I'm Cormac Sheridan, and this week we're taking a look at the progress of a promising therapy in development for amyotrophic lateral sclerosis, or ALS. It's called AP-101, and it has already completed a phase 2 trial. ALS is, of course, a relentless and devastating neurodegenerative disorder. It's caused by the death of motor neurons in the central nervous system and the spinal cord that leads to a loss of control over voluntary muscle movement. Most patients have what's called sporadic ALS, which has no obvious cause, but about 10% of cases arise from a genetic mutation, and upwards of 40 genes have been implicated, which points to the extraordinary complexity of this condition. The type of onset can also vary. Most patients initially experience weakness or twitching in the limbs or hands, although in a minority, the initial symptoms can include difficulties in speaking or swallowing. Either way, the symptoms gain in severity and spread throughout the body, leading eventually to near total paralysis and the need for a feeding tube and for non-invasive ventilation. Although the condition is highly heterogeneous, the disease course is usually quite rapid. Most patients die from respiratory failure within 2 to 5 years of their diagnosis. And despite extensive research and clinical development in the area, progress has been painfully slow. Only 3 drugs have been approved so far, and one of these, Biogen's antisense drug, Qalsody, or tofersen, is only indicated for a small minority of patients with a particular genetic mutation in the gene encoding an enzyme called superoxide dismutase 1. So the vast majority of patients, including those with sporadic disease and those with other familial forms of ALS, have only recourse to 2 options, and neither of them is hugely effective. Riluzole, an inhibitor of the neurotransmitter glutamate, or Radicava, or edaravone, is an antioxidant. But now the Swiss firm AL-S Pharma is the developer of AP-101, and it's among quite a lengthy list of biotech and pharma firms that are in the clinic with investigational agents. The industry pipeline is extraordinarily diverse, which is both a reflection of the huge complexity of the pathophysiology of ALS, but also a reflection of the lack of maybe understanding of what the key disease drivers are in ALS. But ALS Pharma, that is among a minority of firms that has generated promising patient data. We're going to explore that data in this episode with the help of the company's Chief Medical Officer, Angela Genge. Angela, you're very welcome to BioBiz Buzz.
Thank you very much, Cormac.
This isn't your first outing on BioBiz Buzz, of course. Yourself and your colleague, CEO Michael Salzman, you both spoke with my colleague Mike Ward last November. Listeners can check out that episode for more information on the origins of the company, your own professional backgrounds, and on the discovery of AP-101, which is in itself quite a fascinating story. But when I first came across the program and learned that it selectively targets mutated forms of the SOD1 protein, I immediately thought of Biogen's drug Qalsody, or tofersen. There are similarities, but there are also very substantial differences between the two. Can you outline what they are and maybe also explain how their safety profiles differ?
Absolutely. So I'll start with Qalsody just because it's been out in the public sphere. Qalsody targets total SOD1, the normal protein, the production of the normal protein, and works at the level of reducing the production of this normal protein. And by knock-on effect, eventually shows some clinical changes with some biomarker changes in between. But that's its core way of being effective. It is delivered intrathecally, so a spinal tap. And currently you have to receive the drug every month to maintain its benefit. AP-101 is a human monoclonal antibody directed at misfolded SOD1, so the toxic form of SOD1, which occurs in patients with a mutation in their SOD1 gene. But we've demonstrated through our program and, in fact, through other people's research that misfolded SOD1 can occur with wild-type SOD1 and not just mutated SOD1. So there is a population of patients who have sporadic ALS, so non-genetic forms of ALS, whose wild-type SOD1 also misfolds. AP-101, this particular antibody, recognizes only misfolded SOD1, and it recognizes a very specific epitope within the SOD1 protein that is only exposed when SOD1 misfolds. So it never affects total SOD1 or the normal protein at any point in its trajectory.
And the normal protein itself, does that play an important physiological role in patients with ALS?
It plays an important physiologic role in everyone. It's 1% of the protein in the nervous system. It manages oxygen-free radicals and other consequences of oxidative stress in the cells. Motor neurons are large and undergo a lot of activity with the mitochondrial, with the endoplasmic reticulum, and therefore are quite dependent on the normal function of SOD1 to maintain motor neuron health. So the difference from a very basic perspective is we have one molecule that knocks down total SOD1 and another molecule that only binds and clears the misfolded versions.
Does that translate then into differing safety profiles?
The safety profiles of the two molecules are quite different. The intrathecal drug, which is Qalsody, has a list of declared side effects that are followed and watched out for, including inflammatory responses in the cerebrospinal fluid, transverse myelitis, benign intracranial hypertension, and a few other related to infusion of an ASO into the cerebrospinal fluid. The human monoclonal antibody, which is AP-101, actually has a very clean profile. It's given intravenously, not intrathecally, and it has very low rates of any forms of side effects, no significant side effects related to the drug or the drug delivery, and no sort of immune reaction, which is what we always worry about with the use of monoclonal antibodies. There is no injection site reaction, there's no immune reaction, and we've not yet seen any anti-drug antibodies develop with this molecule. So quite safe to date.
Yeah, and to date, I know obviously is always the operative term in an ongoing clinical program. And you have disclosed clinical data, and they are noteworthy from what I could see. I mean, late last year, you reported that it prolonged survival and delayed the need for ventilation, both in patients with sporadic ALS and in patients with SOD1-mutated ALS. I mean, what aspects of the data did you find most encouraging?
So there are a few things, obviously. One is this prolonged survival. But in addition to that, we have a few other key clinical and biomarker results that we think are important. One of the clinical results that are as important is that the effect happens early. And the patients report effect as early as 6 weeks after starting the treatment. We see a delay in progression as measured by 2 or 3 different outcome measures, and particularly in the SOD1 ALS population. We see a maintenance of weight, and in the highest tertile, those patients with sporadic disease that have the highest levels of these misfolded SOD1 proteins, because we have a biomarker assay that can identify the levels of circulating misfolded SOD1 that's recognized AP-101. So the highest tertile, in fact, responds very much like the SOD1 patients. So we have a category of at least a third of patients with sporadic ALS that have high levels of this circulating misfolded SOD1 wild type protein. And when you give them AP-101, they respond like the SOD1 patients. So their weight is maintained, their functional level is maintained. It slows down any loss of function as measured by the ALS-FRSR. So we believe that this is very strong evidence that there is at least a subset of patients with sporadic disease where misfolded SOD1 protein triggering cell death is an important component of their disease.
Okay, that's absolutely fascinating. Because on the composite exploratory endpoint that you employed, even there, the effect size seemed to be greater amongst patients with sporadic disease than amongst patients with SOD1-mutated disease.
Yeah, so when we look at it, break each of the outcome measures down, what we see is this slowing of the loss of function that was most striking when you looked at the ALS-FRS, which is our functional rating scale. in the SOD1 mutant patients treated early and that top tertile of sporadic patients, as very similar results in these two groups. We've also done some biomarker work now that is analyze the biomarkers of the patients at the end of their double-blind period, because their survival benefit is at the end of both the double-blind and then the open-label extension, where even the placebo arm got drug, and we saw the survival benefit. When we look at the biomarkers at the end of the double-blind period, so 6 months in, when you have the longest period of time that there's a difference in treatment between the two, we have statistically significant changes in phosphorylated neurofilament heavy and in neurofilament light. So it suggests that this drug is active and has significant changes in key biomarkers that we now measure as surrogates to a disease progression in ALS. We don't have 12-month data like that because of the open label extension. So that is probably one of the reasons we didn't see this at the end of 12 months is you rescue or improve the situation, even the placebo group. But it was quite striking when we ran our pre-specified, this was all in the plans, pre-specified analyses of these biomarkers and saw a significant difference.
Yeah, and the neurofilament light chain, particularly, I believe, the reductions in that, which is, I understand, a non-selective marker of neuronal damage. It's not specific to ALS. It occurs in several neurodegenerative diseases, as far as I understand. I'm less acquainted with the phosphorylated neurofilament heavy chain. Is that specific for ALS, or is it also a general biomarker of neuronal damage?
Both are general. They have very different characteristics. Part of the reason we talk a lot about neurofilament light is because with the Qalsody program, that was the one thing that changed significantly in their original study. I think you recall that, in fact, they didn't meet their clinical outcome measures at 6 months and had to do a series of analyses and have accelerated approval awaiting some final data on the clinical side. And that rapid response in NFL to Qalsody may be reflective not just of the effect, but may actually really reflect the biology. But the difference between NFL and PNFH is its stability. We know that phosphorylated heavy chain neurofilament is quite a stable form of neurofilament, and so is not affected by things like falls, infections, hitting the head. There's no acute phase reactant type behavior of phosphorylated neurofilament heavy. It is analyzed in the cerebrospinal fluid, and there's data going back 20 years about this in ALS. It is significantly elevated in patients with ALS, and to affect it, you're affecting something over time. So if you bring down your neurofilament, phosphorylated neurofilament heavy, this is indicative of a treatment response. If you follow patients longitudinally, PNFH actually goes up over time, about 0.5 to 1%, if there's no intervention that actually affects the health of the motor neuron. The biggest challenge we're having with NFL in general is that it can change with things that are common in ALS. It's definitely elevated, higher than any other neurodegenerative disease. It is relatively easy to do because it's a blood test. But if they fall, it shoots the number up. If they get an infection, the numbers go up. And that can be problematic when you're trying to use it as a marker of therapeutic benefit. The other thing
It's
is,
an unstable metric, in other words, yeah.
yes, the other thing about NFL is, although we're quite excited about it with Qalsody, we know that there are other programs that take longer than 12 weeks to have an effect on NFL. And one other program that is clear, strong evidence of disease-modifying effect, it is one that is currently being run by Otsuka. It is another antisense oligo supporting patients with bus ALS. We saw people getting out of their wheelchairs and walking, but it took NFL like 9 months to respond. So it could be And this is being looked at right now in real time by scientists who understand the biology of NFL. That we're very happy with the results we have in AL-S Pharma, but we may be asking for a bar set by Qalsody that is much more complex than we currently realize. And so we're happy to see what we have, but we also feel that the story behind neurofilament in ALS may be a bit more complex, but we were able to meet our targets, so we're good.
Yeah. Can anything be drawn in terms of the effect size in the biomarker changes and the associated clinical improvements, or at least slowing of the functional decline in the patients? Is it easy to make those associations, or is the data too kind of messy for that?
Well, the NFL data is messy in everyone's hands.
Yeah.
I think it's not as tight as we first thought, that most development programs are not seeing a very tight time-specific correlation. And we know that the regulators approve drugs based on clinical benefit. And the totality of the evidence, a biomarker can be used as supportive evidence, but at the end of the day, we need to demonstrate clinical benefit, which is what we're seeing in AP-101 and what they're working on with tofersen.
And I know as well, I mean, the company has stated that you plan to initiate a phase 3 trial
Yes.
before year end even, which is
Yeah.
hugely ambitious and very exciting. It's going to attract an awful lot of interest, no doubt about that. And as I was saying earlier, I mean, and as you know better than I do, as a practicing clinician as well as a scientist, ALS is a hugely heterogeneous condition, and there's a huge amount of patient-to-patient diversity. And it's interesting that you did mention that there are at least a subgroup of people in the sporadic cohort who have high levels of misfolded SOD1 protein already present.
Yeah.
Are they a group that you would say preferentially include in the trial, or what are your? I know obviously you haven't published a protocol or probably finalized one yet, but as you go thinking forward about the kind of questions you can approach and how you can approach them, would that be sort of part of the thinking at least?
Oh, it's very, very important, right? The fact that we have a biomarker that we can use at baseline, we can't use it longitudinally because it incorporates AP-101, so that it doesn't line up as a longitudinal biomarker, but it certainly lines up as a way to stratify. So right now, the plan is to stratify, and to stratify based on whether or not you have misfolded SOD1 high levels. And there will be two cohorts, one with mutations and one with sporadic. And within that, we'll stratify high versus medium low levels of misfolded SOD1. And this is actually probably really important because at the end of the day, even with the diverse biology we know exists in ALS, that we can actually target misfolded SOD1 in a significant proportion of sporadics. It actually gives us a runway with this drug and with others to approach this, continuing to better our targets and our understanding. But the biomarker is going to be a key part of stratification. We're working out now the design, obviously, and we will be running the design past the regulators. The regulators to date have wanted us to not limit the number of sporadics based on any criteria, but we do want to stratify because we think that's really important. And it's going to be an international trial. Our phase 2 trial was also dreaming big, and we went to South Korea, North America, and 3 countries in Europe. We'll expand beyond that. For phase 3, we have had a lot of interest, but also we think that this has an opportunity because it is so easy comparatively to give, right?
Yes.
It's a simple IV preparation you give every 3 weeks. It doesn't require any complicated handling. And it's given as an IV, so you don't need the more tertiary care settings to deliver the drug every month. So we think that that is also a real opportunity for this drug.
Yeah, absolutely. And Is there any much information from population studies about what percentage of sporadic patients would have high levels of misfolded wild type SOD1 protein?
So that's a great question. We're the only ones that we know of that have a biomarker assay. We've done a few different cross-sectional studies. We are working to look at several of the researchers in the community that have biobanks longitudinally have asked if we would run our assay on their samples. So when we get to the end of those projects, we'll have at least a larger snapshot of how frequent misfolded SOD1 is in a larger population. So I think that will be very helpful. There's been like 15 years of studies looking at postmortem and a few that are looking at sera and documenting misfolded SOD1, the presence of misfolded SOD1 protein in many different sets of postmortem materials of patients with sporadic ALS. So that goes back since 2010, I think, is one of our benchmarks, where that we've started different groups. It's not just one group. It's actually pretty widespread, have demonstrated this misfolded SOD1 protein in sporadic autopsy specimens. And now we have some results, hoping to build on it, in life patients with misfolded SOD1 circulating.
And yourselves, as well as the whole field, you face the shared problem that the people that you do get to treat, if they have a definitive diagnosis of ALS, it would have more or less taken them a year to arrive at that diagnosis. And their central nervous system has been subjected to this pathological cascade for that time and longer, because it does build before the symptoms are apparent. And it's an awful lot to ask of one drug to do anything. And that's where we're at.
Yes.
What are the hopes for earlier diagnosis and earlier intervention?
Oh, that's a great question. That's a community question, as you know. It takes many forms. People recognizing at the very onset of the disease that this could be something serious. The number of great patient advocacy groups around the world have made it a point of doing basically public announcements on and developing different tools to remind people of ALS. In a very sad way, public figures getting ALS actually seems to shorten the time to diagnosis simply because people think of it. It would be really great to get that 1 year down to 6 months, because that would give us a real fighting chance to make a difference. I can say that this AP-101 molecule, because it works so quickly, and as someone pointed out, probably takes out any circulating toxic proteins in the periphery as well as in the central nervous system, we get this early response that we're not seeing with other therapeutics, which may actually also be really important. But everything we can do to make the world aware of this disease so it's diagnosed early is a big deal. Some of the connections of this disease with sports, in particular with the military, have helped. There's no obvious reason why some actors get it, but there's definitely a long-standing understanding that certain types of physical activities at the professional level increase your risk. And there's a fairly significant effort to understand the connection with the military. But most of this just brings the idea up so people go early.
Yeah. And then maybe a final question that you probably may not be in a position to answer just yet, but the phase 3 data, it's going to take time, obviously, to set up the trial and recruit the trial.
Mm-hmm.
Do you have a rough time scale in terms of when the data might become available?
Oh, you're right. I don't have a great answer. We're very ambitious. We would like to recruit the study within a year of starting, which is not easy and means that we will want to be in a lot of areas in the world. And then it will be another year after that before everyone is done. And so you're talking 2029, potentially. I'm really bad at this kind of math. But, end of 2026 to end of 2027, just to recruit, you've got another year, which is 2028. So that's why I'm saying 2029. I really encourage people with ALS who meet criteria to find a clinical trial to go in. I am a very strong believer that ALS should be treated as any cancer, and every patient should be given the opportunity to enter a clinical trial, because that's how we're going to make a difference. We're the way breast cancer was 30 years ago, and we're just getting our biomarkers now. So this is going to be a trial that we can offer more broadly because of its ease of delivery, and we will be looking at places around the world to run the trial.
Okay. And we'll be watching for those data when they become available with bated breath, although, as you say, it's going to take some time.
Yeah.
And obviously, people have to be patient. But it's going to become a very important study. And I do wish you and your colleagues all the very best of luck with it. ALS, it does continue to place a huge burden on patients and on their carers and on their healthcare systems, and it's crying out for safe and effective therapies. So thank you, Angela, for sharing your thoughts with us. It's been very, very valuable. And I bid you goodbye, and I say goodbye, and thanks everybody else for listening to this episode of BioBiz Buzz. And we welcome your feedback, comments, or thoughts on this or other episodes. So if you want to get in touch, please do log on to biobizbuzz.com. Comment on LinkedIn, Blue Sky, X, or Facebook, and continue listening on Apple Podcasts, Spotify, YouTube, or wherever else you find your podcasts. In the meantime, goodbye for now.